Okay, well, it's great to be here today, and I want to thank Philip Fortier and the Defeat MSA Alliance for inviting me to give this talk. I'm going to talk today about progress towards a biomarker-based diagnosis for multiple system atrophy, maybe a little bit of a misnomer description, but as most people are probably aware, in the last year, we have the result of the task force that came up with the new criteria for diagnosis of multiple system atrophy. And this leads to 3 possible diagnoses shown at the bottom of the screen here. Neuropathologically established multiple system atrophy, which requires microscopic examination of brain tissue, so is mostly meaningful for autopsy, which is not helpful during life. And then 2 categories: clinically established multiple system atrophy, which is based, as clinically established implies, on clinical findings and symptoms, and also includes MRI markers. And then clinically probable multiple system atrophy, which is purely based on clinical findings and symptoms. So the diagnosis of MSA by criteria remains basically a clinical diagnosis. But there are biomarkers, various imaging and laboratory tests, and the task group has addressed this and says that a list of research biomarkers for the multiple system atrophy diagnosis is a supportive investigational tools that provide laboratory findings suggestive of MSA, but it's not required for the diagnosis. And table 4 lists these biomarkers. They are not formally included in the MDS MSA criteria. The bold and italics are mine, and that's because they're not available to everyone to use. Some of these tests are fairly limited in their availability. Others have perhaps not always the diagnostic accuracy we'd like, and in many cases, we just don't have enough data to validate them as diagnostic tools. Nonetheless, the report goes on to say that they recommend using research biomarkers at all levels of diagnostic categories as much as possible. And basically, this will generate more data about how useful these biomarkers are and how they may become included in the diagnostic criteria. And the task force, again, the emphasis is mine, will plan the systematic identification of emerging or consolidating diagnostic biomarkers to promptly advise the update of MSA criteria. So going forward, biomarkers will hopefully have an increasing role in how we establish the diagnosis. But I want to talk particularly about some biomarkers that may be directed more at the actual etiology, the causes of the disease, and how that may help us. And so if we go to the next slide, this is the table 4 referenced before. I think Dr. Lang may be doing a talk about the rest of the criteria, but we're just going to focus on table 4 here. And there are MRI markers, as alluded to earlier, and looking for atrophy and other changes, visible changes in different areas of the brain. There's FGG PET biomarkers, a different type of imaging that shows metabolism. And so you look for reduced metabolism in certain areas of the brain associated with either multiple system atrophy type P, Parkinsonian, or multiple system atrophy C, the cerebellar form. Then there's normal cardiac sympathetic imaging. Why normal? Well, because regular Parkinson's disease is known to cause abnormal sympathetic innervation of the heart. And so cardiac sympathetic imaging will pick this up in cases of regular Parkinson's, and a normal scan will make it more likely that you're dealing with multiple system atrophy or some other Parkinsonian variant. PSG proven REM sleep behavior disorder. Over recent years, it's been very clear that during rapid eye movement sleep, which is when you dream, people with these Parkinsonian diseases have a tendency to act out what they're dreaming. PSG is a polysomnogram and will show that there is REM sleep without atony. So the normal person will sleep, but have very little movement during their dreams because their motor system goes into neutral, if you will, and they don't act out their dreams. Whereas patients with Parkinson's disease, multiple system atrophy, a variety of other synucleinopathies, as we call them, and we'll get to that in a moment, tend to not disengage the motor system and end up acting out their sleep and have behavior during their dreams. Another test is supine plasma norepinephrine. One cause of neurogenic orthostatic hypotension is if you have a problem with the peripheral nerves that innervate the blood vessels and release norepinephrine to tense up the blood vessels and maintain blood pressure as you change position to standing. If you're losing those nerves to the blood vessels, then there's less norepinephrine available. And so you tend to get very low levels of supine plasma norepinephrine. So if there's greater than 100 picograms per mil, that's telling us, okay, your problem with orthostatic hypotension is not coming from the peripheral nerves, it's coming presumably from the brain, and makes it more likely to be multiple system atrophy. I'm going to skip through some of these and not explain all of them in detail, and skip down to cerebral spinal fluid, CSF, alpha-synuclein oligomers detected by PMCA or RT-QuIC. And this is where the concept of synucleinopathies come in, and this is actual detection of abnormal aggregates of a protein called alpha-synuclein in the spinal fluid. There's another CSF biochemical test mentioned here. I'm not going to talk about that. Really is an indication of a breakdown of certain nerves in the nervous system. So moving towards the non-clinical diagnosis, most of these biomarkers, as you'll have recognized from my description, are based on effects of the disease. The brain imaging showing atrophy in specific regions, the FDG PET showing decreased metabolism in different regions of the brain. The cardiac sympathetic imaging detecting that there's not loss of sympathetic nerves in the heart. The serum plasma norepinephrine levels indicating that there's not loss of nerve fibers causing the orthostatic hypotension. However, the newer biomarkers are based on the hypothesized cause of the disease, the abnormal alpha-synuclein oligomers or aggregates that can be detected in spinal fluid and elsewhere. And this is different because we're looking at the cause of the disease rather than the effects of the disease, although the idea that the abnormal alpha-synuclein folding and aggregation is truly causative is not totally accepted. And this goes back about 20 years when Dr. Brock and his colleagues looked at a large series of autopsy cases in Parkinson's disease and came up with this staging. And as you can see in the diagrams of the brain, where the darker areas are the more affected by the pathology of Parkinson's, the Lewy body pathology. It's starting in the brainstem, the darkest areas, and spreading and gradually spreading throughout the brain. And the little table on the left reflects this idea that as the disease progresses, you get more pathology and the places involved first, and spreading of the pathology to different parts of the brain. And this, not too many years, I guess about 5 years later, was brought into more focus by the discovery that patients who had been experimentally treated for Parkinson's disease by having tissue that had intact dopaminergic neurons implanted into their brains eventually died and went to autopsy, and it was discovered that this Lewy body pathology that was a sign of the Parkinson's had spread from the patient's brain into the normal tissue that was transplanted into the brain. So people began thinking in terms of the pathology actually spreading from one part of the brain to the other and into otherwise normal tissue that was not expected to develop Parkinson's. And so a whole series of various experimental reports here mentioning that you could take exogenous alpha-synuclein fibrils, again, in aggregate form, and induce dysfunction in neurons and neuron death by just applying them to nervous system tissue, that there's a phosphorylation event that controls the toxicity and the aggregation in a particular model of animal model of Parkinson's, and more this idea that there's cell-to-cell transmission of pathogenic proteins in the neurodegenerative diseases, not just Parkinson's, but also in multiple system atrophy, in other Parkinsonian diseases, and some other diseases that aren't Parkinsonian, including Alzheimer's disease. And this led to what's called a Prionoid mechanism. So you see at the top of the screen, this little colorful squiggle that's supposed to represent the protein chain. Proteins are made as little strings of amino acids, sort of like a little bead chain, but they have to fold up to make a three-dimensional shape in order to be functional in the cell. So you could imagine, for instance, that the red block that's just over from the left side of the screen is the functional alpha-synuclein folded in the appropriate way, but it could fold in some other ways, and we actually know that the body has ways of getting rid of some proteins that just happen to fold abnormally. But the interesting feature of some proteins is you see on the outside on either side, the possibility of folding into a prion-like conformation. And what this means is that once it folds into that shape, it can clump together, and you see it clumping together into bigger and bigger aggregates going down the sides of the figure on either side. And that not only does it form these clumps, but the presence of the abnormally folded protein seems to induce other protein of the same type to fold in order to bind to the aggregate. So here you have a mechanism where you develop these aggregates of misfolded protein that can induce more of the protein to misfold. And the idea in the Prionoid mechanism is that then that protein can spread from cell to cell and introduce the same misfolding into otherwise normal cells, and hence the spreading that we see through the brain in the Brock hypothesis, and the spreading from Parkinson's brain to implanted normal tissue that we saw in the graft patients. And so these aggregates then form the Lewy bodies and some other pathologic structures, which we recognize as the hallmark of certain Parkinsonian diseases, particularly true Parkinson's disease, and therefore are suspected to be the cause of the disease process. Maybe not the big aggregates like the Lewy bodies, but maybe the smaller aggregates as they're forming can interfere potentially, and there's quite a bit of evidence, with the function of certain cells and cause dysfunction and even cell death. So we move on. We know that abnormally folded protein can induce more abnormal folding and aggregation. So here, spinal fluid from patients has been put in a test tube with additional alpha-synuclein or other suitable substrate, and basically gently shaken for a long period of time, and aggregates form. And so you see in the healthy controls, HC to the far left, no aggregation. In the tubes with spinal fluid that was taken from multiple system atrophy patients, there's the blue dots representing one form of aggregation that occurs. And in the PD, Parkinson's disease spinal fluids, there's a different form of aggregation with a different level of fluorescence. And if you look at the panel B, it shows that the speed, the kinetics of how these aggregates form also differs. And there are actually a number of other tests that can be done to show that they are in fact the kind of aggregates that form in the brain and are related to the Lewy body pathology. And because you can see this distinction, now you'll notice that the red and blue are provided to indicate what type of patient the spinal fluid was taken from. But you see a few blue dots that are up there where most of the orange dots are. And they may actually be patients who really do have Parkinson's and are just thought to have multiple system atrophy, one of those things that has often frustrated us and leads to the reason for these stringent diagnostic criteria. And conversely, there's a bunch of orange dots that are down at the blue level that may well be patients who are thought to have Parkinson's disease, but really have multiple system atrophy, but haven't been established to have that diagnosis yet. But nonetheless, you can certainly distinguish different types of synucleinopathies in terms of different folding, different type of aggregates being formed based on spinal fluid taken from patients. And certainly any of this aggregation indicates what we now call a synucleinopathy. And this goes a little further. In order to not have the complications of misdiagnosis, these are similar sorts of test tube measurements of aggregation, but using brain tissue from autopsies that has been homogenized and used as the seeding factor to start the aggregation in the test tube. And so we have here frontal cortex and substantia nigra compacta tissue taken from patients who have pathologically proven dementia with Lewy bodies, DLB, versus patients with pathologically proven Parkinson's disease, and controls who do not have any pathologic evidence for a synucleinopathy in their autopsy brains. And again, you can see there's a specific kinetics, a specific type of aggregation that is triggered by seeding with abnormal aggregates from patients who had dementia with Lewy bodies. And it's not all about alpha-synuclein. I mentioned Alzheimer's disease earlier. There's a different protein called tau, which can misfold and induce aggregation in a similar prionoid fashion. And so we have tauopathies. And so on the left side of the screen, we have a graph where there are a bunch of orange dots representing aggregates formed from Alzheimer's disease, and then comparing the aggregates formed in the same assay using a number of other diseases, including similar to Alzheimer's. We have the chronic traumatic encephalopathy, but then we move into PSP, another Parkinsonian type disease that's in the differential diagnosis and the confounder for do you have MSA, do you have Parkinson's. We have CBD, cortical basilar degeneration, another Parkinson's plus disease. And then we have some other things like Pick's disease and frontotemporal dementias. And you can see the conditions here, the vertical lines, they tried inducing aggregates from tissue from an animal where they knocked out the tau. So the animal has no tau, you get no aggregation or very little, just spontaneous aggregation of the substrate in the test tube. The blue vertical line is the average for tissue taken from non-tauopathy patients. And then the other, the colored dots indicating how much aggregation. And this particular substrate, the R306, is particularly good for inducing further aggregation caused by Alzheimer's disease. And in the other graph to the right, we have yet a different substrate that in similar fashion was used to pull out the Pick's disease patients as being separate aggregation from what they saw with Alzheimer's and PSP, progressive supranuclear palsy, cortical basilar degeneration, and so forth. So it's possible to do these aggregation studies on at least two different types of protein that are associated with different diagnoses that are in the differential diagnosis for multiple system atrophy and for Parkinson's disease. And I point out with progressive supranuclear palsy, PSP, which is a tauopathy, but while there's the typical in red PSP, there are a variety of types of subtypes of progressive supranuclear palsy, including some PSPP, looks mostly like Parkinson's, PSP-CBS looks mostly like cortical basilar disease, PSPC gives cerebellar ataxia and may look somewhat like multiple system atrophy. And then, of course, we know in multiple system atrophy, we have both the cerebellar and the Parkinsonian type. So this makes for a lot of the difficulty in clinically sorting patients to specific diagnoses. So do we actually have this biomarker to use? Well, sort of. So this is data for Syntap, which is a commercially available test to be done on spinal fluid to demonstrate synucleinopathy. I see I dropped an L in synuclein, but the point is here, if you look on the left, the Syntap biomarker, they either did or did not detect abnormal alpha-synuclein aggregates. And then going across the three columns, they expected to detect it in patients with clinically diagnosed Parkinsonian syndromes, most of them probably true Parkinson's, confirmed by having a positive DAT scan. So they had definite loss of dopaminergic neurons in the brain. Expected result of not detected, these were spinal fluid from a large number of patients, 109, that were not known to have any neurologic disease. And you can see where you, in the top left corner, detected 48 patients who were expected to have a synucleinopathy. Actually, the test detected that they did have a synucleinopathy. But there were 7 patients out of the 55 that did not get detected, even though they were expected to have a synucleinopathy. on the not detected column in the middle, only 3 patients had a spontaneously abnormal test, despite not having known neurologic disease. So less than 3%. Most of those patients, 106 patients, they didn't detect it and they didn't expect to detect it. And of course, there are the confounding situations. Some of the expected detection that was not detected might actually represent a tauopathy patient with a Parkinsonian form like the PSP or the cortical basilar degeneration. And the 3 patients where they detected abnormal alpha-synuclein, even though they did not have no neurologic disease, might have had very early Parkinsonian disease. So, nothing's ever 100%, but then in the lower part of the chart, you can see the percentages for how sensitive the test is, how specific it is, overall accuracy. It's pretty good at detecting synucleinopathy. When they do this test, they see something of the kinetics of formation, and they suspect they may see differences between patients with true Parkinson's and multiple system atrophy, but they're not able to reliably report that at this point. So this test just proves that you do in fact have abnormal alpha-synuclein aggregates in your spinal fluid, but not which disease you have. Another place that you can see abnormal alpha-synuclein aggregating is in the skin. And you can actually, I mentioned grinding of brain tissue to be the seeding of the aggregation studies in the test tubes. That's been done with skin biopsies as well. And you can see, again, the propagation of aggregation in the test tube that's triggered by the abnormal alpha-synuclein found in the skin. But you can also take a skin biopsy and do immunostaining under a microscope, which is what's being done here. So the top row, the A panels, are from multiple system atrophy patients, actually a patient for this particular skin biopsy. And the top left, the green PGP, this is a general marker that fluoresces, the green in this case, for nerve fibers of all sorts. And they're looking at the superficial dermis, the upper part of the skin. And there's a particular area where there's a subepidermal plexi. And you see the asterisk in that panel that reflects the depth into the skin where those plexi are. In A prime, you see the red phosphorylated synuclein that's forming aggregates, a little clump of it pointed to by the arrow. And it's at that level in the skin. And in the final panel, a double prime, so to speak, where they're merging both the PGP and the phosphorylated synuclein images. You can see that the clump of red stuff is in fact in some of the green nerve fibers, and it's at the level of the subepidermal plexus. In contrast, in the bottom row, B through B double prime, we're seeing a Parkinson's patient with orthostatic hypotension. And here the deposits are found in autonomic nerve fibers deep in the dermis. Actually, this small blood vessel here. The green in this case is staining for tyrosine hydroxylase. So it specifically is marking adrenergic nerve fibers. There may be other nerve fibers present. And then the red is again where there is phosphorylated alpha-synuclein clumping, and you merge them together, and you can see again that the aggregates of abnormal alpha-synuclein seem to be associated with the autonomic nerve fibers in this part of the deeper part of the skin. So again, we have available, you can send off a skin biopsy to be tested to see whether there's abnormal alpha-synuclein aggregates in it, but we do not yet have the ability to reliably say, oh, well, these aggregates are in areas that are associated with multiple system atrophy as opposed to these others are in areas that are associated with Parkinson's disease, though this particular set of images would suggest that that should be possible, but it will take a lot more samples and data before we can really say, yes, you can distinguish these diagnoses by where in the skin you find the abnormally aggregated alpha-synuclein. But we're headed that way, that between the test tube assays of how the protein aggregates and what kind of seeding of abnormally folded alpha-synuclein may be present in spinal fluid, brain, skin, and the immunostaining of the skin biopsies where the abnormally folded alpha-synuclein, whether it's present and where it's present in what structures of the nervous system innervating the skin, may be able in the future to actually distinguish which type of abnormal folding is going on. So this leads us to our conclusions. One, we have tests available today that can tell from spinal fluid or from skin biopsy whether a synucleinopathy is present or not, with not absolute certainty, but with some pretty good certainty. So this can help us. And certainly, if you don't find synucleinopathy in a patient that you suspect has Parkinson's or multiple system atrophy, you might want to look and see, could they really actually have a tauopathy instead, and so forth. But the available tests can't, or have not been proven to differentiate between the different types of synucleinopathies, such as Parkinson's disease, multiple system atrophy, and dementia with Lewy bodies. Although the research data that I showed you certainly suggests that that will be possible. So this differentiation may soon be possible, giving us diagnoses of PD type aggregation versus MSA type aggregation versus DLB type aggregation. Now, we mentioned earlier that there's a lot of clinical variation in the clinically diagnosed diseases, even when you look later at autopsy and have a firm pathologic diagnosis. Some people's clinical presentation and course doesn't really suggest the disease that they actually have, or is a very atypical form of it. And so this means that very likely will be patients whose clinical diagnosis is still different from their aggregation type. Somewhat speculative, but I think it's almost certainly going to be true. And even more speculative, but seems very likely, is that drugs that are developed to prevent and disrupt the synuclein aggregation, we believe that that's a major cause of the pathology and nervous system tissue loss that causes the symptoms that reflect the disease. If we could block the aggregation, we can slow down and potentially even stop the spread of this pathology through the brain. And we have drugs that are even now in clinical trials to try and prove that they can interfere with synuclein aggregation and slow the progression of multiple system atrophy. They very likely will preferentially target specific aggregation types. So this has two sides to it. One is, if you're testing a drug, you'd like to test it in a group of patients who all definitely have the same aggregation type. It will give you a better chance of proving that the drug actually works. But then also, when you go to treat people with these drugs, you're going to want to use a drug that seems to really target the aggregation type that's present in that patient. So a patient with variant Parkinson's that looks like maybe MSA, but really has Parkinson's type aggregation, you're going to want to treat with a Parkinson's type aggregation drug. A patient who seems to have variant MSA, but really actually has you know, Parkinson's aggregation, you're going to want to use that kind of drug target. So we're going to ideally come down to better treatments and more discussion about what actually constitutes a diagnosis. Is it important to know the clinical descriptor of the disease, or is it important to know the underlying pathology? So we're going to be seeing more excitement as the biomarkers become better established and understood, and more changes in the diagnosis and treatment of the disease, which is very promising. So I want to thank you for your attention, and it's really great to have so many people attending virtually this conference. And I want again to thank the organizers.